Time to move over to our next speaker, which is also the last speaker for this session and for the virtual summit. And I believe we had a little bit of a mistake on one of our slides early on, where Stephen Bourne was actually affiliated to Merck. So anybody who picked up on it, and I think Stephen has, just to acknowledge that we made a mistake here. This is not the third Merck talk that we have in our summit. We already had two yesterday. So Stephen is in fact actually from Continuous Pharmaceuticals in the US, and Continuous Pharmaceuticals is a startup out of MIT and Novartis, which has really taken the whole approach to bring continuous manufacturing from raw materials to final product going out of the door of a pharmaceutical manufacturing company, really to the next level. So what Continuous Pharmaceuticals is looking into is an end-to-end solution, not only the chemical synthesis, but everything that happens downstream as well. As many of you know, the last step of the API synthesis, making pharmaceuticals, is not where manufacturing of these drugs end. There's a lot of steps that come afterwards, including crystallization, purification of the API that needs to happen before it can be administered as a drug, filtration steps, and finally compounding and putting it into a form so the consumer can use it, if it's a tablet or if it's a capsule, or or other more complicated ways of administering drugs, such as sprays and others. And Continuous Pharmaceuticals really had looked into a lot of these processes as well, and turning them, what was usually or quite often is classically done still under batch conditions, turning that into a continuous process. So a few more words maybe on Stephen as well. Stephen is the Director of Scientific Affairs at Continuous Pharmaceuticals. Before he joined Continuous, he was a postdoctoral research fellow at MIT under none other than Professor Klaus Jensen in the Continuous Flow Chemistry Center at MIT. He's now responsible for innovation, for identifying and developing new unit operations and integrating them into the continuous manufacturing processes for small molecule pharmaceuticals. He's also works closely together with the FDA's emerging technology team. And we're very much looking forward to hearing from you, Stephen, this morning about your latest developments at Continuous. I have to say a very big thank you also before you start for getting up this very early. I know it is only, I believe, a quarter to six where you're joining us from the US. So thank you very much for rising so early, and I hope you're fueled up on coffee. And over to you.
Thank you so much. Let me share my screen. Can you hear me okay?
All good, yes. Coming through clear.
All right, and can you see my screen?
Screen is also coming up nicely. Thank you, Stephen.
Thank you. Come on, you. There we go. All right, so thank you all for the invitation, and thank you also to the other speakers. It was really great to hear of all the awesome work that you're doing, and thank you, Christian, for moderating this session. So I'll just jump right into my talk. This slide I'd like to show you is a slide that I stole from the FDA. And this, I don't think they mind sharing it. It's their vision for pharmaceutical quality for the 21st century. And that is an agile, flexible pharmaceutical manufacturing sector that reliably produces high quality drugs without extensive regulatory oversight. And so they're interested in any way to improve quality and also to address supply chain issues and everything that usually pharmaceutical companies get a hit for if something screws up. So they like to encourage new technologies. Continuous manufacturing is used in just about every other major industry in the world except for the pharmaceutical industry. As was previously said, it's a very conservative industry and very slow to change and highly regulated. So every success that we do kind of reverberates through, and we're helping to bring this change. And we think that continuous manufacturing can really improve product quality. So when we at Continuous think about all things continuous, we see things in this kind of chart here, where you see this risk versus reward, where you see greater reward when you go continuous. So you could think of it like an individual unit operation. Clearly, from all the flow chemistry examples you see with improvements in heat transfer and mass transfer, you can really overcome a lot of limitations that chemistry has. And you start to see more benefits when you integrate additional unit ops together. So when we get to point B, where you can say integrate all of the steps, all of the synthetic steps for making a drug substance or all of the steps for making a drug product, you see a lot of benefits there. There's also more risk in terms of time, money, and concern about regulatory hurdles. And then there's the most reward when you get all the way to a fully end-to-end integrated continuous manufacturing system where you're feeding raw materials in one end and you're getting finished product out the other. So processes that usually take lead times of 2 years can be done in as quick as days, which just from a supply chain perspective in this current world we live in is very meaningful and also a very good business case. So the way that we see this is you can think of it as much as you all think about continuous flow as a seamless process. Here we just kind of include the scope of it to include not just the chemistry, but also producing the API and also doing your formulation to be able to produce your drug product form. So to achieve this continuous flow, of course, end-to-end integration, so integrating different unit operations like reaction with extraction, with crystallization, filtration, and so forth, taking a big picture system approach. So thinking about all the different solvents you might use and maybe trying to simplify the solvents to a more uniform solvent or just reducing the number of different solvents. It really helps at simplifying that and making a more robust manufacturing platform. Also, none of this would happen if you didn't have an integrated control strategy. So you could think of it like what Svetlana was referring to in the case of using a lab manager to be able to record data. That's very important, especially if you could have feedback control loops for the data that you're collecting to be able to provide feed-forward feedback control to maintain the homeostasis of your process. In the absence of that, it's almost like a Rube Goldberg machine where anything can go wrong. So to jump into a case, and as you can see, this is a slide that chemical engineers like, but we had a case where we had to produce an API, and fortunately, this is an API that was produced in a single step. For the chemists in the crowd, it was a CN bond forming reaction. I'll just leave it at that. Literally A plus B gives you C. Now, as a departure from Svetlana's talk, where everything runs in a tubular reactor, this is a case of flow chemistry that no one really wants to do. This is not a fast reaction. This is a reaction that has solids. So again, DMF reflux, 10 to 14 hours of residence time. This is a reaction that produces solids, and a lot of solids. This is a highly concentrated process stream that has over 78% of solids in it. Product C crystallizes out before full conversion, so you can't run it to full conversion in solution and then crystallize it out separately. It starts to crystallize out within like an hour. So, no amount of tubular reactors won't save you in this case. Like Svetlana's, it's a highly exothermic reaction, and the crystallization is also highly exothermic. And also this product, just for information, it's a high volume generic, and we were asked to design a line that would produce over 10,000 metric tons a year. And I'm not kidding, this is one of the top 5 produced drugs in human history in terms of mass, and it's not acetaminophen, it's not ibuprofen, it's not aspirin, but it's up there. And so the client is already running this, I think, at 12,000 metric tons a year, and really because of the exothermic nature of this reaction, they could not scale it up in batch anymore, just because of the thermally generated side reaction materials. They couldn't purge that to spec in subsequent filtration. So they were really looking at anything that could help them meet this huge unmet need of supply. And so working with them and thinking, it'd be nice to fit it in a tubular reactor, and quickly realizing that that doesn't really happen, we had to think differently. So this is the footprint of the pilot plant where this work took place. So just for reference, this box on the left next to the gray box, those are 2 back-to-back 2-meter fume hoods. So these are bathrooms, but so here we have a series of process enclosures where we landed equipment for each of these unit operations. So the first enclosure is a dissolution enclosure where we added the raw materials from these bag dump stations here and here. These were pneumatically conveyed, one of them milled and pneumatically conveyed into gravimetric feeders, which fed into a dissolution vessel. One of the raw materials was a little, had some insoluble impurities that needed to be removed. So that pre-reaction mixture solution went through a continuous polish filtration that we designed and developed. And then the resulting pre-reaction mixture went into a CSTR cascade for the reaction, 5-stage CSTR cascade where the reaction and crystallization happen. Subsequently, we filtered the material on our continuous filtration system and then resuspended it and then fed that into our dryer, our continuous dryer, again, something that we also had to develop ourselves. This material, the dry API that has, at this point, we used PAT to measure all of the critical quality attributes. So everything was in spec for the API, was pneumatically conveyed to the next enclosure, which for EMC, it stands for extrusion, molding, and coating, where we're able to make tablets. And I'll show you that very briefly. And then an additional thing, because this is a high volume generic drug and every cost matters, the filtrate that we collect and the solvent vapor that we condense, we have a solvent recycling and recovery unit here where we use thin film evaporators and distillation columns to continuously distill, purify, and enable us to reuse the two solvents. One of them was DMF and the other one was IPA. The CTO cabinet is the control system. The operator station is here. And as you can see from these dimensions, it's about 9 meters by, I don't know, like say 4.5 meters in terms of size and dimensions. So here's a PAT overview and a process flow diagram. Again, the raw material feeds come in. They go into a dissolution vessel. We clarify it through a continuous polish filtration. We have our 5-stage CSTR cascade. In the last vessel, we land some PAT for process understanding, but not for measuring any critical quality attributes. We go through our continuous filter, and then we resuspend the wet cake. This enables us to monitor the main impurity down to ppm level real time. We then feed this into our continuous dryer, it's a drum dryer, and this material, once upon drying, goes through an airlock where we're able to measure residual solvent content by NIR, confirm the crystal form by Raman, and also use laser diffraction to confirm the to measure the particle size. That was another specification for that. The material then was fed into a twin screw melt extruder where we were feeding in an excipient blend of the inactive ingredients. The blending step, which are what extruders do, was able to mix this blend to the correct content uniformity, which was measured by NIR. And because this is a hot melt extrusion and we wanted to maintain the crystallinity of our API, we had an inline Raman to be able to confirm the crystal form of what we were producing. And then we just injection mold tablets on the back end of that. So again, this is what the pilot plant looks like from the front end. And so here you can see these CSTRs. These are 10-liter jacketed vessels. Again, it looks like a batch vessel, but you can actually run these in series. So you can handle solids and flow and slurries and flow. And these were all connected to recirculating heat exchangers and peristaltic pumps. So again, looking a bit more deep dive into it, we received our raw materials in 25 kilo bags, and these were pneumatically conveyed into these pneumatic receivers, and then drop on call into these gravimetric feeders, which meter A and B with the correct stoichiometry into a 20-liter jacketed vessel, which we're also feeding in DMF as the process solvent. At 78%, remarkably, this solid goes into solution at 80 degrees C, and the reaction doesn't happen yet, which is important. We continuously polish filter this stream. We want to remove that ash, that insoluble impurity in the process stream, because going into the reaction and crystallization, you cannot filter that out afterwards. So it's important to get it out beforehand. So here, these are just your simple, tea filters. I think Svetlana is using something similar. And again, it's important to measure the pressure across these. So as the pressure goes up, what we did was we had two in parallel, and so when the pressure got up to a certain level that we knew we were entering a nonlinear region of pressure increase, we would switch to automatically switch to the clean and readied filter element. And then this compromised one, now isolated, could be backwashed, cleaned in place, and prepped fully automated. So that way we can run these just back and forth, and we can have continuous polyps filtration. And this was done, they would automatically trip for at least this process about every day and a half. So again, it's a 5-stage CSTR cascade. With PAT, we have mid-IR and FBRM in the 5th stage. This is, these are fancy pictures of silica and water for the Italian film crew when they took pictures of this. In reality, when you're running at 78% concentration, for this process, it looked like a hot, corrosive milkshake. And these pictures look prettier, but you can imagine it's just a thick thing that would never flow through a tubular reactor. I wish it would flow through a tubular reactor, but this was a viable, robust solution that scales very well for the residence time and the conditions that we were looking at. So for continuous filtration, so this is what it looks like from the bird's eye view. So it consists of a porous metal plate, and here comes the process stream. We pump here. We have an oscillating motor here, which just helps us get a good spread of slurry along the radius of the plate. This plate rotates clockwise, and along these radii, we have wash blocks. So we have a vacuum that pulls, it's just vacuum filtration. We have a vacuum that pulls the mother liquor through, and these wash blocks here wash clean solvent onto the wet cake, displacing the mother liquor and purifying it. Here's another view of it where you can see these wash blocks. You can heat the wash blocks if you want. And this wet cake would get removed through the combined action of a scraper blade and auger. This would then get conveyed into a resuspension vessel. And we do this for a couple reasons. One, it enables us to, well, for regulatory purposes, if you did a thing, you have to prove you did it. And so this is a purification step, and we want to measure the purity of our API. So we landed some React-IR probe here to measure the amount of impurity that was still in that mixture with respect to the API. Second reason, this enables us to, our dryer operates on a thin film evaporation principle, a lot like a thin film evaporator or a rotavap. And so it works better to convey material as a slurry, and also our dryer works better if it's a slurry instead of just a pasty cake. So again, the residence time on this filter is typically about a minute, 1 to 2 minutes, and you can change that, and we have just in terms of PAT for monitoring this, we have pressure sensors underneath the filter plate that help us monitor if you clog your filter, and all filters will foul. Your pressure, your vacuum pressure will get stronger. And so to address this on this other radii, after we remove the wet cake from the plate, we can continuously wash the clean plate before slurry gets added. So that way the incoming slurry always sees fresh, clean plate, and we can have a robust unit operation of filtration that doesn't clog over days and days and days. So the next unit up is our continuous drum dryer. And so you can see 2 stainless steel drums, they co-rotate inward. Here's the slurry being applied to the middle. And the residence time in this is about a minute, sometimes less. And it dries it to ppm amount less than the spec, or within spec for all of the solvents that we put through it. So the drums are electrically heated on the inside, and then the chamber that it's in, we can place it under vacuum. So much like a rotavap where you can control temperature and also put it under vacuum, we get very efficient evaporation. The material upon drying flows through and drops, goes through a cyclonic separator, drops through an airlock, and that gives us an opportunity to use PAT to measure the residual solvent content of the API by NIR, the particle size distribution using a Malvern Incitec system, and also the crystal form by Raman. So I'll just move forward the next steps very briefly, but the extrusion molding coating system, we call it 3-in-1 product manufacturing. Essentially, you have a gravimetric feeder where you can feed in your API into this. You've got your excipient, which then comes into this here. You can see it feeds into this hopper. A twin screw melt extruder, it's kind of like the way that pasta or Play-Doh is made. And it will blend the excipient with the API to give you the right content uniformity, and then we injection mold tablets. These are conveniently dyed our company's color, orange. and it puts out tablets. And the residence time through this is about 10 minutes. So very quick all the way through. And so it's just important to be able to balance this all out when you're kind of viewing a systems approach to this. So at this point, I'd like to pivot. And so that was a process development lab, and this is a manufacturing facility that we are currently building out right now. And so what you see here, what exists right now is this 4-building as a warm shell. We haven't built the utility building behind it yet. Currently we're located in Massachusetts, and rather than outsourcing our manufacturing to another country, we're building this manufacturing facility. It's about a mile from our current R&D labs. And so from the back view, you can see here we've got storage tanks for solvents. There's a lot of other utilities in here. So in addition to us being able to produce API in this facility, we're also producing drug product as well, since we like everything integrated, not just tablets, but also sterile injectables too. So that would be sterile injectable solutions in vials. And so we recently this year, thanks to the pandemic, we received a large US government contract to produce 3 APIs and integrating that with formulation, fill, and finish for vials. And so these 3 APIs are very interesting. So they're all sterile injectables, and 2 of them are highly potent, and 1 of them is a controlled substance. So that's made our R&D life a little interesting, and also going through the whole permitting process. We just got our DEA license, which is exciting and also scary. And so just to kind of describe some of the chemistries very briefly that we'll be doing for the manufacturing of these 3 APIs. There's acylation and alkylation. We have a continuous Grignard formation in use. There's a condensation reaction. We're doing asymmetric hydrogenations in a PET bed. Kind of like there was a really nice paper from AstraZeneca where they did this in OPRD. And so we're kind of doing it in that fashion. Reductive aminations and also dynamic kinetic resolutions as well. So with that, there are some additional things that we needed to develop. If you recall that filter that you saw, you can look into the filter. And because now we're going to be working with highly potent materials, there are 2 things that are very important when working with highly potent materials: engineering controls, as in like a closed system that doesn't contaminate everything else around it. And so that's 1. And 2, the ability to clean in place. That's very important. And I'm going to take a page out of Svetlana's book about doing riboflavin testing for proving your cleaning validation. That's important because you don't want your highly potent controlled substance to be getting into your other drugs in a multi-product production facility. So again, this is just a CAD picture of our rotary filter for the manufacturing, our drum dryer. So again, 2 drums, looks fairly similar, just kind of looks like it's on steroids. And then these are placed in an isolator. So that way, when you're generating, you know, 1 million doses an hour, for every couple hours worth of highly potent substance, you can be safe and protected from it. So all of this is just very important, and how we're growing as a company. With that, so we've accomplished this end-to-end integrated continuous manufacturing at the R&D pilot scale, and we've advanced this with potent compound capability. Working with the US government and the Department of Defense and Health and Human Services, we're implementing the first US multi-product full CGMP factory, which will be ready in 2023. And we'll be able to manufacture and sell essential generic medicines, as well as provide CDMO services for innovator drug companies. So with that, I want to thank you for your attention, and I look forward to any questions you might have. So thank you.
Thank you very much, Stephen. That was truly a fascinating talk, seeing how you integrate all these individual unit operations, all these processing steps in a really, truly end-to-end continuous production facility, production capability. I'm just sending out another request to everybody online that's still listening to us. If you have any questions, please fire them through. And here they come. So there's a first question coming in from Dipali Arora. What level of control was obtained over the crystal morphology? Was there any particle aggregation observed in flow, and what product quality control was implemented? It's a 3-in-1.
Yeah, that's a good question. So in general, we want to control crystal form, crystal size, crystal shape. In this case, because it's we were constrained by the economics of this process. When people run this, there is no control over crystal shape or crystal size. When you run at this scale for such highly exothermic and concentrated conditions, you're running at such high supersaturation that it's kind of ridiculous. There's no control over it. The only thing you can do is you can stir fast enough in these vessels, and it's done at the manufacturing scale, that you don't get incrustation. However, I think you get to a certain point where you're running so concentrated that the bulk mass of your process mixture actually kind of wipes the insides of your crystallizers kind of clean. So it's kind of self-anti-fouling to a certain degree. But yeah, so we did not do anything. It's a robust process, so it makes the right crystal form. As far as the particle size and shape, one thing that I did not include was we went through an inline wet mill in between the filter and the dryer, so we can hit our particle size spec. And again, we monitor that real time with FBRM in the wet mill vessel, as well as after we've dried the API, it goes through a dry-based laser particle size flow cell. For control, you could do feedback control for that, but with the right unit operations like a wet mill in place, we were able to hit spec all of the time. It wasn't an issue.
Thank you, Stephen. I've got one from Volker Hessel that's kind of linked to what you just described around the particle size. He says also, great talk. The compounding equipment is usually specific for certain particle sizes. Does your continuous method allow for a wide range or all particle sizes to be addressed, or are there limitations?
That's a great question. So particle sizes is the bane of anybody working in drug product formulation for tablets. Just it all comes down to the flowability and the ability of being able to mix them well. It's like trying to mix sand and gravel. No matter how much you shake it, you can't mix it right. For us, what works really well is you're not doing much powder handling. Pneumatically conveying is easy no matter what the particle size. Gravimetrically feeding is great no matter what the particle size. And so if you feed these into a hopper for our melt extruder that's with a steep enough angle, they'll all flow in well enough. And as the excipients melt, the viscosity ends up improving the mixing, facilitating the mixing of this with the twin screws. So we can work, like particle size is not an issue. We're not powder blending, we're feeding powders in, but it's a melt formulation where we maintain the crystallinity of the API and have the excipient blend melt.
Thank you. Michael Young wants to know again what the maximum production capabilities in like kilograms per hour is of the pilot plant.
Good question. So the nominal flow rate for this was about 1.5 kilo an hour of API. We built flexibility in it to be able to double the throughput or reduce it by half and still be able to produce in-spec material. So the most we ever took it up to was about 3 kilo an hour throughput. That was the design criteria for our filter and dryer systems. Yep. And again, that's unique to this API because we're running this at like 70%. So I would say what would be more normal for a pilot plant of this type. Again, we're not running GMP in this pilot plant, so the more you put through it, you can put more through it. But what purpose does it have? If it's not going to be something that you can sell or put into people, then it's useless. So my perspective is you should make one, you should operate everything in continuous flow so you can capture the dynamics for that, and two, operate at an appropriate scale-up factor between what you want to do from the bench to what your manufacturing throughput should be. But a lot of people think like, how much can you do? And it's not about how much you can do. It's about, at the pilot plant level, it's about exploring the development and design space. You want to explore variation. That is the antithesis of manufacturing. In manufacturing, you want to eliminate all variation. So I don't think a pilot plant is appropriate for manufacturing. It's more information gathering, and then that ensures that your manufacturing build out and your manufacturing process is as boring as possible, because that means that it's producing exactly what you want, when you want, all the time.
You mentioned GMP, and that's a question from my end that I had, and I think a lot of us working in process development and process R&D in general, I get that question answered in one form or another by clients all the time. You said your pilot plant is not running under GMP, but obviously the plans for the final production facilities are. At what stage are you considering changing over to a GMP facilities? Is it past pilot plan generally, or where do you, and what are you asked to do quite often by your clients?
So so after our pilot plant, we run GMP. One thing in our manufacturing facility is that a lot of the projects that we receive from clients, here's a good example of the 3 drugs. So the U.S. government has some weird asks, especially in the case of a pandemic. Operation Warp Speed told them, like, if companies could make 100 million doses of vaccine, that would be great. When it came for asking companies to produce hydroxychloroquine, they asked 100 million doses. That would be great. For this project, for this contract, they asked for 100 million doses of capability of each of these 3 drugs. When you do the math and you figure out what that means, given the dosage size, one of them was 5,000 kilos a year, and the other 2, the highly potent ones, were 15 and 20 kilos a year. So there's a huge difference in throughput. So that in that API facility, we have a high volume suite and we have a low volume suite. And so that low volume suite is equivalent to the size of our pilot plant. It's just, so you could consider that a pilot plant, but that's going to be in a GMP environment.
There's still more questions coming in. I don't know if you have the time to answer all of them. I'll pick one from Tash Polisos, who's asking, or he's talking about the economics. So obviously that's an important factor for generic API manufacturing. How important is this in establishing onshore manufacturing for generics?
It's big. I mean, really, the pharmaceutical industry has been outsourcing manufacturing to China and India for the last 40 plus years because labor's cheap. And so you can't really expect that to happen overnight. And ultimately, as far as, onshore manufacturing, you need to come up with a cost competitive way to do it. And so I think that continuous manufacturing and flow chemistry is the way to do that. You have a much lower in-process volume, which means your equipment is much smaller. As a consequence, you have more control over it. And that gives you typically, and I think everybody here has probably encountered it at one time or another, higher yield, cleaner reaction profiles, which means that your separations, your purification is easier. And so you can make the case, you can make the economic case for it. As far as where it is, I think, in terms of supply chains, and that's a current topic, if it's in your country, it's easier to get. So, will this happen for all drugs? Probably not. But I think in terms of national strategy for essential medicines, that you probably want to produce some, onshore, just so that way you don't have a lack of them. But for other ones, you have to make a case on it. You might be able to, like what Roche does is stockpile a bunch of API. In the case of Tamiflu, which is a pandemic flu drug for the swine and bird flu variety. So, that's a long lead time, and to prevent any kind of kinks in the supply chain for that, they have a large finished supply of API stockpiled. So in case there's a pandemic, they can meet it. Too bad Tamiflu doesn't work for COVID, but that's something that Roche has found works. And so, yeah, I think you can make an economic case for it, but it's important to do that.
That's very interesting, and a topic that obviously is becoming more and more important over the last few years. I know that we are already at the end of our time. I might throw in maybe just one last one with a title request for maybe a short answer, Stephen. One last question from Bert. He's asking, how do you handle hiccups in the equipment train, like short pump problems, sudden stop of the filter or others?
Have sensors in place that tell you what your flow rates are. So if something goes down, the operator can know this, have maybe backup parts. So that way you can do a quick drop in to fix it. And then the other thing is to build in surge tanks. So, if you've got a CSTR that has a certain fixed capacity, that buys you time, it buys you hours. And for these small systems, that's really, you don't need much time to, replace a pump. But yeah, those three things help a lot.
Thank you. Thank you very much again, Stephen. Thank you for a fascinating talk, and thank you especially for getting up that early in your day. I know it's very late in our day for anybody here joining in Australia. If I can persuade all of you to just spare another few more minutes, I would like to share a few slides that I've prepared earlier about the upcoming IMRET, obviously next year. So I'm just bringing up my my screen, and I hope everybody can see this. So thank you first and foremost. So with the last talk we just heard from Stephen, this really concludes this first off, we don't know, virtual IMRET that we held over the last 2 days. I have to say a very big thank you on behalf of the whole organizing committee to all of our speakers and to all of our panelists that joined us yesterday and today. We had some fantastic talks, truly fantastic talks from industry throughout the globe. Really, there was from pharmaceutical manufacturing to bulk chemical manufacturing, quite a broad bandwidth that we heard, all using new flow chemistry or continuous processing methods of one form or another. Starting off with Sebastian Hadner and Christian Holze from Merck and BASF, talking about modular process concepts and the holistic approach that BASF is taking to flow chemistry, introducing things like decentralized manufacturing processes in their manufacturing. Then in the second half, yesterday we heard from Bert Metten, from René Lebel, and from Cecilia Bottecchia how certain examples are transferred into flow in their respective production facilities at Ajinomoto, Roche, and at Merck Sharp Dome. Really fantastic talks about how to make diazomethane safely and much more efficiently with much less waste compared to a batch process. René talked about what they're doing in the area of fenebrutinib, again making pharmaceuticals using 3D printed catalysts, and then again a great talk from Cecilia using or showing us their current developments on pilot plant scale using photochemistry and many others. And then today we had again a great lineup of speakers, starting with Oliver Hutt, who unfortunately couldn't join us live, but we saw him on the video. So thank you to Oliver for sharing the latest development at Boron Molecular, how Boron is building new capabilities in flow chemistry manufacturing. And then we heard from Peter Peckelhauer, fantastic talk about digital twins and the latest developments at Thermo Fisher. And then the last two talks in this session from Lana and from Stephen that we just heard. Again, great talks on how to use 3D printed reactor technology to handle very difficult cryogenic processes using organolithium. And a fantastic talk from Stephen just now on the modular concept that they're employing to end-to-end pharmaceutical manufacturing using continuous flow. A big thank you also to our panelists that joined us earlier today, Paul Watts and Joshua Boyle from Boron Molecular. Big thanks to Joshua for doing that also on very short notice and last minute. And last but not least, a very big thank you to Caroline McMillan for moderating that fantastic panel discussion. So thank you to all, everyone who has contributed. thank you to all the other panel members, and a big thank you to Roger Stableford from the Royal Australian Chemical Institute for organizing today. I've got a few very short slides on what we have in store for next year. So speaking on behalf of the whole organizing committee, we'd like to invite you to join us next year for a real live, in-person, face-to-face, good old conference here in Melbourne. So IMRET 16 will be held from 27th to the 30th of November at the Melbourne Convention Exhibition Centre. And for all of you joining us from the Northern Hemisphere, for us, 27th to 30th of November is the start of our summer, or early or late spring. So the time right now is actually a very good time. If you've never traveled to Australia, if you've never traveled to Melbourne, it's a very good time to come down under and join us for this really great event. If you have been here before, you possibly know. If you haven't, I would give the advice to pack both light clothing and also maybe a rain cover, because Melbourne can be quite unpredictable at that time of year. Right now we have fantastic weather, and I hope this continues over the next few days. Melbourne is the second largest city in Australia, and if you, let's say, bring your swimming gear as well, there is a fantastic coastline that can be discovered just a short drive away from Melbourne. There's pictures here up on the top, which is from the 12 Apostles on the Great Ocean Road. So there's lots of scenery to do if you were planning to stay for a few days before or after the conference. Very quick and a few words on the venue itself. So very fortunate that we could get our conference into the Melbourne Convention Exhibition Centre, and we had, many of you might know that we had started planning with them already back in 2019 for IMRET 16 to actually be held at the convention center in 2020. It's meant to be around about a year ago from now, in December last year. But then, as we all know, COVID happened, and we had to postpone it twice now. But with the new dates set for 27th to 30th of November next year, we're very confident that by then, international travel will have continued to normalize, and we would be looking forward to joining all of you, seeing you in the Melbourne Convention Exhibition Centre. Actually, the last time I've been in there, only a few weeks ago, was for very different purpose. The last two times I've been there, I had my COVID vaccination in the MCEC, as it was possibly the largest vaccination center that we have here in Melbourne, and that took up larger parts of this year. So as we, I guess, going into 2022, we believe that everything will normalize again, and they will be getting lots of conventions and exhibitions into the center. On the bottom here, you see some of the facilities that we have. So IMRET 16 will have a range of different parallel sessions. We've got large auditorium rooms, and we will also have an exhibition floor. So if anybody's interested to exhibit at IMRET 16, please get in contact with us. Please get in contact with the Royal Australian Chemical Institute, who will also be organizing next year's event. And one last slide, very briefly, on our plenary and keynote speakers and our themes that we had already organized for IMRET 16 next year. One of the upsides of COVID was that we had an extremely long planning period for IMRET 16. We had already started to plan for this conference back in 2018. And that gave us obviously also the time to look very carefully on who we wanted to invite and who we could get into Melbourne for as an academic speaker. And here's a long list of the plenaries and keynotes that are already confirmed. I'm only going to very briefly mention the first three speakers, which are plenaries. Oliver Kappe from the University of Graz, Jana Stoudemire will be talking about her latest developments and work at Axiom Space and how flow chemistry is important to them and their business. And then we will also have one of our own locals here from Monash University, Tanja Junkers, who will give one of the plenaries as well. In terms of the themes, it's a very broad spectrum of different themes for IMRET 16. So there'll be classical topics such as microfluidics and microreactor development and development and new systems. There'll be themes that speak to the pharma sector, the fine chemicals manufacturing, but also more recent emerging or recently developing or recently getting more attention type applications, such as in the energy space, using microreactors for CO2 utilization or for hydrogen. But as I already mentioned, there'll be also quite some exotic or in growing and in emerging markets, such as flow chemistry applications in space or in other remote locations. There will be, again, coverage of various different processing methods using electrochemistry, using photochemistry, applications in organic synthesis and polymer synthesis. And lastly, of course, we'll be looking into process scale-up and industrial applications, but also into more newer developments in the introduction of artificial intelligence and machine learning, and the combination with the flow chemistry methods that are currently employed. So with that, I'll leave you with this image and with a final message from myself, thanking everyone who's joined us today across the globe. I know we've got participants from a range of different time zones, and I would like to thank you and welcome you if you're planning to come to Australia next year for IMRET 16. And I know it's been said before today, I know I'm not going to say these same words when we have the real in-life IMRET next year, but for now I'd like to say good morning, good afternoon, and good night.